we are amazing beings, and we're amazing for lots of reasons. But one of the things that I'm particularly interested about is the fact that we're so complex in how we're made up. The different type of cells that we're made of, and the sheer number of cells that we're made of. Can anyone hazard a guess as to how many cells there might be in an average human body? I can't hear you.
3 million.
3 million. Higher or lower? Higher. Right. Where are we up to next? Trillions. Okay, so that's much closer to the truth.
But not
Okay.
as many as there are of bacteria.
That's true. Yes. Far inferior to the number of bacteria. 50 trillion cells in the human body. Okay? It's an extraordinary number, isn't it? Absolutely extraordinary. And it's stated there are about 200 different cell types. Actually, that's probably not true. There's probably a whole, much bigger range of different cell types than we can even possibly imagine, but nevertheless dwarfed by the total number of cells in the human body. Again, perhaps another really amazing thing about all of these cells, which are obviously microscopic, is that every single one of them contains DNA. Okay, and of course the DNA is the stuff that makes us us. It's the things that encode genes, about 30,000 genes that are active, and those genes make the building blocks, the proteins that constitute our cells. And if you were to take all of the DNA in our cells and put them end to end, how far do you think that would reach? Back to the sun and back?
Yes.
Yes. More than that, okay? Oops. That's how long it is. 66 round trips to the sun. Okay? Isn't that extraordinary? That's all packaged up, okay, into a single human being. Okay? So that's how complex we are. That's how amazing we are. And perhaps that's one of the reasons we generate the diversity of cell types and the complexity.
I'm really worried that you're going to step back on the little box behind you.
This one, I'm going to trip. Yes, I'll kick it out of the way. Thank you. I think it's an amazing thing that we are constituted in this way, and in fact, that we work at all. You imagine carrying that kind of machinery around you, and having to, repair it like you would your washing machine. I mean, it would be never-ending, wouldn't it? And yet it works, and it works fantastically well. So, this is why I study the human body, and this is why I'm interested in disease processes, because we're starting off with such amazing things. Okay, so stem cells then. What are stem cells? Well, of course, stem cells have got, slightly out of order, but the stem cells are the things that we're all come from. So those trillions of cells start off, would you believe, with just one cell. Okay, and that cell will divide and divide again, and as it divides, it becomes more and more specialized, and it becomes the adult cells that we know about. But that stem cell has got two characteristics. One, because it needs to be able to generate that many different number of cells, it's got to be able to renew itself. Okay, so the ability to self-renew endlessly, a kind of immortality, is one of the features of stem cells. The other feature of a stem cell is the ability to turn into any one of these cell types. And we call that pluripotency. So that stem cell could become a muscle cell, a nerve cell, a heart cell, a kidney cell. And this is what constitutes a stem cell. So when we're trying to decide whether or not what we've got in our hands, in our labs, in the dish, is a stem cell, we're looking for those two types of properties. Okay. Does anyone know who this is? I heard a yes and I heard a no. Okay, are we going to hear who this is? So this is Prometheus. Okay, and Prometheus was a titan who is credited with a variety of different things, but the one that he's being punished for is bringing fire to humans, and therefore bringing civilization. And for that, Zeus chains him to a rock for eternity. And every day, an eagle comes and pecks out and eats the liver of this titan. But of course, the liver grows back. Okay? It regenerates. And that is actually something that the liver can do. The liver can regenerate. So this is an early example of stem cell medicine. Okay? And what's extraordinary is that actually many of the tissues that we have in the human body have got stem cells in them, defined by the fact that they can self-renew, at least to some degree, and they can become a number of different types of cells. And they can vary in how many different types of cells that they have, that they can become. And we often, rather than calling it pluripotency, we'll call it multipotency. So it's slightly more restricted. But we see these type of stem cells in muscles, in bone and in fat. In the brain, there are certain areas, and in the skin. And we think these adult tissue stem cells are important in maintaining the homeostasis of tissues, keeping them viable, renewing them when required. But you all know that, if I remove, 90% of your brain, it's not going to grow back. Okay, so they don't have a full capacity to be able to do that. They maintain a degree of homeostasis, but it's far from complete. And other animals, nevertheless, for example, if you take the zebrafish, they've got much more pronounced abilities to regenerate, and that's inspired a lot of hope for people with cardiac disorders, because one of the features of the zebrafish heart is that it can fully regenerate. And I'll come back to this a little bit later, but there are hopes that we can learn from that and remodel and regenerate our heart cells. Okay, but these are a type of stem cell, tissue stem cells. Let's go back to the question of what makes a cell. And I said every single one of our cells has got DNA. But there's a problem with that, isn't there? Because if what determines a cell is the DNA, and in fact every single cell has got exactly the same DNA, how do you get all the different cell types? How does one cell know to be different from another cell? So perhaps, as this cover suggests, we're not entirely determined by our DNA. We're determined by a number of other things, but another key factor that's involved is something called epigenetics. And that turns out to be really important in cell identity. What is epigenetics? Well, epigenetics are modifications of your DNA code, of the 30,000 genes and of the regulatory factors that drive them, and it determines within a particular situation whether a set of genes are on or a set of genes are off. And that's beautifully encapsulated in the metamorphosis that a caterpillar undergoes. Same set of DNA, and yet very different organisms at different stages of its life cycle. And it's all down to epigenetics. So it's effectively an on-off switch for your genes. And these modifications, as I say, turn out to be really important for determining cell identity. And so we know that the epigenetic marks from a skin cell is very different to the epigenetic marks of a liver cell. And that's part of the reason why they're two very different types of cells. Okay. So this is the process that we've already gone through from getting from a single cell to an adult. Can we turn the clock back? Okay, can we reverse that clock? Can we reverse the process? Well, it turns out that you can. Absolutely extraordinary that there was discovered, not so long ago, a process called reprogramming, which can absolutely turn that clock back from an adult cell into that more primitive state of a stem cell with the ability to self-renew and the ability to become any of the cell types in the body. Okay? Now, you would have thought that was quite hard. That'd be a quite hard thing to do. It was quite hard. It took quite a long time to get there and to work out what it was that was needed to get these cells back, to reprogram them. And it was discovered by this chap, Yamanaka, in 2006. And it turned out that only 4 factors, a cocktail of just 4 factors, is the secret to immortality, is the secret to reversing the clock. And by adding these cocktail of 4 factors to any adult cell, it could be a hair cell, it could be a blood sample, a skin sample, you can turn the clock back and make an embryonic-like stem cell. And we call it, because it's being reprogrammed, induced pluripotent stem cells. Okay? And this built upon the work of John Gurdon, who did a lot of that early work here in Oxford, then went off to Cambridge and is currently still working in Cambridge. And this discovery was so revolutionary, and it has absolutely transformed life sciences, that very fittingly, and a very short time later, after the discovery, they were given the Nobel Prize. Okay? So this is another type of stem cell that very commonly we work in the lab, the human-induced pluripotent stem cell. So what we have then are a number of different cell types, a number of different stem cell types, and these include the tissue stem cells, which are resident in adult tissue, embryonic stem cells, which are in embryos and have been experimented upon. Again, lots of ethical issues around that. And more recently, the induced pluripotent stem cell. Okay. So I'm going to stop a little bit with the stem cells, and now I'm going to turn to disease and why it is that these type of technologies are important for us right now. So one of the things that's been happening over the last several decades is that the disorders that we've been fighting with and the ones that we've been really concentrating on are those disorders that kill us, and very rightly so. And what's happening is that, the big conditions like ischemic heart disease, so in other words, heart attacks, strokes, cancers, these are the big things that kill us. And the medicines that we have are starting to make an impact. But because of the changing demographics of our society, and because of the therapies that are coming through, a lot of the public health measures in particular, vaccines, that burden of disease is changing. So we can see global deaths falling, but what's happening is that we're starting to become afflicted by disorders that affect the quality of our life, that impart disability that we're living with. So what we're starting to appreciate is that we don't really have treatments and therapies for living well. So we can prolong our lives, we can live until 80, 90, beyond, but that segment of our lives isn't necessarily at the best quality, and there's been a real paucity of research at addressing those kind of needs. And you can see the kind of disorders that we're really dealing with are pain disorders, psychiatric disorders, diabetes, and so on. And these are the main things that people, if you look at, add up all the disability in the world, these are the big ticket items. And yet, for very many of them, we don't have any useful therapies that really take some things away. A very scary one that's really up on the up, it's small print at the moment, but it's ever growing, is Alzheimer's disease. Okay? Huge problem that's going to be facing us. It's going to have huge impacts upon our society. And do we have a single meaningful therapy for Alzheimer's disease? We don't. There's nothing that will make a difference to the underlying disease process. Okay? So how can we find drugs for these kind of disorders? Well, where do drugs come from? Well, a lot of the drugs, in fact some of the best drugs, like aspirin, have been around forever. Okay, folklore, the willow bark source of aspirin. And many of those kind of medicines are still with us today and are really very effective. A lot of the other drugs come through serendipity. So penicillin is a good example. Not a very good experimenter, left their petri dishes out on the window shelf and, got contaminated. But it turned out, penicillin came from that. Viagra is a really recent example, developed for high blood pressure. It completely bombed. Didn't make any money at all. And then people who were taking it noticed an interesting side effect. And that made Pfizer $2 billion and growing. Okay, so it's, chance plays a big role. And then there's the rational process, which is what, we'd like to sell you as being the way that we're discovering drugs. Okay, we can study the disease process, we understand it, we know what the molecular drivers are, and then we can design drugs specifically for that. Does that work? Well, it does. Okay, but what you can see is that actually, all in all, the rate at which we're discovering drugs isn't that huge. Okay, roughly between 30, 40 new drugs every year. And how much does one drug cost to make? Okay, $1 billion minimum. Okay, and so can you imagine that? The amount of investment that's needed is going up and up and up, and it's not making an impact on the number of drugs that are coming through. This is a huge crisis for pharma. Huge crisis. Really don't know what to do and how to make things better. Okay, but maybe part of the problem is that when they're going through the drug discovery process, they're using methods which don't really get at the human condition. So when you're trying to understand disease and when you're trying to discover drugs, you need model systems. And the kind of model systems that we use are things like immortalized cell lines. These are cancer cell lines. Or it might be animal models. And we discover disease processes and drugs using those systems. And of course, by the time any of those compounds get through to the clinic, a lot of investment's already gone in, and it's all been in model systems that are distant to humans. And so is it surprising that we don't find that it translates across into humans? So perhaps the models we're using are not representative enough. And for the same reason, we can't predict whether a drug is going to be toxic. Is it going to cause liver failure? Is it going to cause, renal failure? So this is perhaps a problem with the current process. But that's potentially where stem cells could come in. Because what stem cells allows you to do is to obtain a sample directly from a person with that condition. And you then are able to interrogate, because you can get the stem cell from, say, a skin sample from that patient, and turn it into a cell that's affected by the disease process. And so for the first time, what you have in your hands to experiment with are cells that are human, that are directly from the patient that is suffering with that disease process. And that's revolutionary for someone like me who's a neurologist. I can't ask my patients to donate their nerves when they come and visit me in clinic. Okay, I'm not going to be able to open up their brains. Okay, but by this technology, for the first time, I get access to their nerves. Okay? So that's one of the reasons why this technology is so powerful and why we are getting so excited by it. So what are the kind of things that we can do with the cells? So we can make, as I said, neurons, and we can compare that against neurons that we might be obtaining from surgical samples and seeing how representative our cells are compared to those directly that we're obtaining. And we can see what the properties of those nerve cells are like. We can use clever tools. These are called optogenetics. And these are clever ways of probing the function and directing the activity of the cells that we're growing in the dish. We can make more complicated models like the blood-brain barrier, the barrier that exists between our peripheral circulation and the brain itself. There's a very strong barrier that protects our brain. And we can find ways of modeling that in our hands. And we can even start to make mini brains in the dish. Isn't that extraordinary? Okay, called cerebral organoids. It's a long way away from a real brain, but we're starting to get at the complexity of real tissues in real humans. And as I say, this is from the patients that are suffering with the condition. So this gives you a way of, you might have heard the term personalized medicine. This is real personalized medicine because you're really dealing with the patient's own cells. Okay? So these are the kind of things that we can then start to do. We start with the patients, we make the stem cells, we turn them into our cells that we think are affected by the disease process. We look and see what the abnormalities are in the dish, and then we put compounds on and see whether we can correct that abnormality. Because if the compound corrects the abnormality, then maybe that's also going to be effective for the patient when you give it to them. So that's the kind of grand scheme of what it is my group in particular are trying to do. And my group's working with a big consortium of multinational pharma companies to try to make this a reality. And I come back to the fact that these are, for the first time, human cells acting in a much more physiological way than the artificial systems that we would have had before. And importantly, they have all of the genes and the DNA that the patient has, and also, therefore, the predispositions, the genetic predispositions that the patient has. I have to still emphasize, this is still, nevertheless, an artificial system. It's still a model. Okay? At the end of the day, these are isolated cells that are existing in a dish, and that's very different to something in the human body. But nevertheless, by having these cells in a dish, it allows us to get through lots and lots of experiments and start to understand the disease process and start to screen drugs. Okay, so what about headache then? Okay, what on earth am I going to do with headache and cells? Okay, so as I say, I run the Oxford Headache Service. Migraine is a hugely disabling disorder. It's actually ranked as one of the leading causes of disability worldwide. Many people don't appreciate that. It's sort of more prevalent than asthma, epilepsy, diabetes all combined. And although there are drugs out there, about 3% of the adult population, so millions of people, do not have medicines that work for them. Okay. And the problem to try to understand this disorder is that it's really complicated. Clinically, there's lots of different subtypes, there's lots of different things that might be sort of underlying it. And so we investigate the genetics, the environmental influences, hormones, and so on that might be predisposing it. We investigate the brain mechanisms and the vascular mechanisms that might be involved in migraine. And what we're trying to do is to distill that down into this model in a dish, which we can then do what I said before. Okay, so can nerves in a dish develop migraine? They can't, obviously. Okay, how can you tell whether a nerve can develop migraine? But what you can look at are properties of those nerve cells that might be a marker, that might be related to a migraine feature. Now, one of the things that we've learnt from all of the other studies is that pain nerves in migraine are more excitable. And so that's the kind of abnormality that we might be looking for in our dish. And in fact, these are real traces, okay, of our stem cells turned into pain nerves. And you can see that in a healthy person, not very excitable, you don't get very many what we call action potentials, spikes of signals, whereas in 2 patients that we've looked at, it's much more excitable. You get repetitive firing of the nerves. So these are examples of phenotypes abnormalities that we see in the cells in a dish that we can then relate to the overall disease process. And so what we try to do then is to apply drugs and see whether we can move from that hyperexcitability to something that's a bit more stable. So that's what we're trying to do. Okay. All sounds easy, doesn't it? Okay. But it's not that easy, and it's not that easy because what I've shown you are where the disease is being caused by a very strong mutation. And so if there's a very strong mutation, then we know that when we grow the cells in the dish, that there'll be very obvious abnormalities in the cells. Now, if you take a more complex condition like Alzheimer's disease or Parkinson's disease, where it isn't a single gene that's causing it, but there's a lot of different genes that are causing it, then the abnormalities in the dish suddenly become much more subtle. And the problem is that they start to get lost in the noise. There's a lot of variability in the cultures that we're making because it's a long and complicated process. There are lots of factors that can come together that can influence your readout. And so that signal, as it diminishes, it gets lost in that noise. That's one problem. The second problem is, even if when we start to find abnormalities, because now there isn't such a very strong gene that's driving it and we can pin it on that gene function, it's much more diffuse than that. When we see an abnormality, how do we know whether or not that's actually related to the disease? Okay, so that's another really big problem. So for example, if in an Alzheimer's neuron that you were growing, it shriveled up and sort of degenerated, that you probably will say, yeah, that's pretty good. Fortunately, they often don't do that. Often it's just something much more subtle that's occurring. And then the question is how do you pick that out and show that that's related to the disease? So we're making really good progress with these very strong gene abnormality disorders, but we're a long way away from doing something very meaningful with the more complex conditions, with the more prevalent conditions. But that's where we want to get to. Okay. So that's what I've talked about so far, is disease modeling and drug discovery. And I would say that that's actually a closer to coming to reality than what I'm now going to talk about, which is regenerative medicine. And there are a number of different ways of thinking about regenerative medicine and a number of different approaches. I can't go through all of them. But one of the ways that I think perhaps might be most likely to succeed is coming back to that ability of cells and the fact that they contain stem cells within them. So perhaps what you can do is think about the things that would reactivate those stem cells and get those to start repairing the damage that might have occurred. And so this is, for example, a colleague of mine in Oxford, Paul Riley, who is working on exactly this. Can you think about how to reactivate the cardiac stem cells, the progenitor cells, so that, let's say you have a heart attack and you've got damage, you can then come in and cause the damaged heart cells to be repaired, okay, to be replaced. And that, I think, in the heart is probably closer to reality than other areas, but there's no reason why you couldn't do the same thing with brain neural stem cells. And in fact, what actually is a reality right now and very tractable is in hematological disorders, where again there are hemopoietic stem cells that can be reactivated and used to repopulate after you've got a problem with a blood disorder. Okay? So this, in terms of regenerative medicine, I feel is closer to being achieved. What about regenerating the human brain? And I mean, there's a lot of companies out there that are selling to desperate people who have incurable conditions and offering stem cells as a way of being able to regenerate nerve cells that are lost. And it's sad to say, for the most part, that there is absolutely no evidence that it can provide any benefit. Why might that be? Well, this is an example of a mouse brain, and that's a small area called the hippocampus, which turns out to be really important for memory. I don't think it projects very well, but these are all of the number of cells that are in that structure. That's a teeny tiny structure, okay, within the mouse brain. Okay, now what about the human brain? This is a representation of not the number of cells, but just the number of connections between one cell and another. Okay, the human brain is extraordinarily complex. 100 billion neurons, okay, is the kind of figures that are quoted. And when you have a disease process, and even if the disease process is only affecting a small proportion of the brain, and then you're saying, I'm going to deliver stem cells and I'm going to regenerate that brain for you, I mean, that's really not feasible, not in the current technologies that we have. But there is no shortage. I mean, if you look on the internet now and you type this up, there's no shortage of clinics that are offering stem cells with exactly that stated objective. And it is utter nonsense. Okay. One area where there may be hope that something could come through are disorders where the disease burden is much more focused, and that's something that may be true of Parkinson's disease. In fact, it is a disorder that affects lots of different systems, but we know a big brunt of the disorder is on an area of the brain called the substantia nigra. And that's a key part that controls your movement, and we know that nerve cells in that area are lost early. So one of the things that one can do, therefore, is directly transplant progenitor cells into that area. And people have initially been investigating fetal transplants, and there are reasons why, which I don't have time to get into, why stem cell-based therapies are a bit more challenging. But if there's going to be an area that's going to work for a central neurological disorder, I think Parkinson's disease is going to be one of them. The reasons why it's really difficult to know whether these things work or not is because as scientists and as clinicians, to be satisfied that we believe that a particular therapy is effective, we like to undertake clinical trials. And we like to undertake clinical trials where we're blind and the patient is blind to the treatment that they're getting. So that stops any bias. Because you can imagine the placebo effect of having a brain transplant is going to be pretty huge. You go and have a brain transplant, you're really going to believe that's going to make you better. And so this is the problem for when you're trying to think about whether or not a transplant really works or not. Because the placebo effect is unbelievably strong. And you can get huge changes in your performance just from the placebo effect alone. And so the question is your real transplant actually having any more of an effect than just the placebo? And getting ethics to do a brain transplant where you're having a placebo is pretty tough, right? I mean, how many people are going to volunteer for a trial like that? But actually, that's what really needed to be done, and that's what they're actually doing. Okay? And I think they're due to report very soon on the outcomes of that. And as difficult as those trials are, that's really what you have to do if you're going to show that these kind of therapies, which potentially are quite hazardous, are going to work. And this is the kind of things that, the BRC is all about, supporting those kind of clinical trials. Regenerative medicine for Alzheimer's, for the reasons I've touched on, is a lot harder. But of course, it's what everybody's thinking about and trying to do. And we can see the amount of shrinkage that occurs from a healthy brain to an Alzheimer's brain, and we can see the kind of pathology that occurs. And again, the kind of things that you'll see is, let's take some stem cells, and it will regenerate that back into a full-on brain. Okay? This is from a company that's currently live on the internet. Okay? And that is patently not possible and is not going to happen. because that complexity is just not going to be repopulated. But again, what might happen and what might be feasible is this ability to be able to reactivate the neural stem cells that are there, the blue cells, and get them to target to the areas of damage. And it might not be able to recreate the full brain, but at least it might be able to slow the process. And I think those type of trials are more attractive, yeah.
There's another thing I've heard of recently, which is not so sophisticated as any of these. There's a US company which is selling pills that will encourage your stem cells to regenerate themselves. So this contains leucine, L-carnosine, and blueberry extract.
It's the blueberry that does it, clearly.
It's supposed to increase the number of CD34 plus cells.
So they're talking about peripheral blood stem cells, and I don't know about that, but I'd be very sceptical that there is evidence that that actually does, what it purports to. What we know, so it's similar to what they're describing, what I'm describing here. It's reactivating resident stem cells. And the reason it's going to be more complicated than that is because the more people investigate how these cells exist in human tissues, they realize it's a really complicated microenvironment. There's, we call it a niche, a stem cell niche, and that's true for every resident adult tissue stem cell. And small changes in the way that niche is behaving is as important as modulating the cells themselves. And so that cocktail of three factors, even with the blueberry, I suspect is not going to be enough to be able to do what they suggest that they're going to do. But as a kind of concept of reactivating resident stem cells, I think that does have promise. I think we still need the evidence, but as a concept, I think it's an attractive one. There are lots of trials going on, though. So despite the challenges, it is a very, very active area of research. And this is just a sketch of the kind of things that we're using, not we, collectively, globally, a type of stem cell called a mesenchymal stem cell that, for example, you can obtain from fat or from bone marrow. And you can see all of the different disease indications, all of the different areas that this is being used for. And these are the different type of trials. Phase 1 is really early safety trials. Phase 2 are kind of the smaller, does it really work? And then phase 3 is proper testing out whether it works for that particular disease. And so you're starting to see these cell therapies starting to come into clinical trials and into practice. How do these work? They're not going to be regenerating tissues, but what they might be doing is modulating the immune system or the area that's damaged in order to be able to help with preventing disease progression, and perhaps that they're working. These therapies have progressed into humans because they've shown promise in animal models. So when they've tried it in animal models, they've shown promise. To date, I've yet to be convinced that a fully well-designed clinical trial has shown an efficacy of this type of mesenchymal stem cells. There are other examples which have worked. As I say, blood disorders, I think, is a good example. Eye disorders, I think, would be another early win for this type of approach. But what's encouraging, I think, is that these kind of things are coming through. And that's also true with neural stem cells as well. Again, providing neural stem cells into disorders such as stroke, motor neuron disease, spinal cord injury. And again, you can see the numbers of patients that are involved are quite small. These kind of therapies and trials are really expensive, so you can't conduct big studies. But this is where we're starting, and I think it really is encouraging that we're seeing these kind of things. But there's many, many years ahead, I think, for this kind of therapy. And there's still lots of challenges ahead to overcome to make this a reality. But I think the promise is there. I think there are excellent people working on this, trying to solve these problems. And one of the key things, of course, is that a lot of this research is only possible because of the excellent involvement of the community to this research. The consortium that I lead, we're recruiting 500 subjects. We've done about 450 or so now for the studies that we're doing, and that only was possible. Most of those were recruited from Oxford. That was only possible because of the overwhelming generosity of the people, that we see in clinics and who want to help, even though they know for them it's not going to directly help them. But they're there knowing that it will help future generations. So at that, I'll end, and thank you very much.